In this video, we are going to look at rotary motors. There are two classic examples of rotary motors. The first one is the F0, F1 ATPAs. A classic example of a rotary motor. The second example of a rotary motor is the flagella. This particular video, let us focus on the flagellar motor. Somewhere in the towards the end of 19th century, a scientist called Wilhelm Pfeffer, he carried out an experiment. What he did was a very simple experiment. He took capillary tubes, and in the capillary tube, he filled it up with a sugar solution. And this was introduced to a medium containing bacteria. So, what he observed was, if it is a sugar solution, bacteria got accumulated near the capillary end. He did another experiment, that is, replace the sugar solution with phenol or any substance which is not an attractant for a bacteria. And he found that bacteria got dispelled. They moved away from the tip of the capillary. So, which is in a way indicating that bacteria are getting attracted towards a chemical attractant, and they repel when there is a repellent is present. So, this process is what is known as the chemotaxis. Now, this chemotaxis was studied elaborately in the 1960s by a scientist known as Julius Adler. In 1960, he studied this phenomenon elaborately. So, what he realized is there are about 20 substances, 20 receptors are present in a bacteria. And these 20 receptors, we can pull them under two categories. One is for attractant, one set of receptors for attractant, another set of receptors for repellent. So these receptors which are present in the plasma membrane as well as in the periplasmic space are soluble protein, which will help to sense if the surrounding medium is an attractant or a repellent. Depending on that, the bacteria will move. What we are going to discuss today is, we are not going to look at the mechanism, the signaling mechanism which is leading towards this movement. We will focus on the motor aspect of a bacteria. That is our point. If you look at the movement of bacteria, so the movement of bacteria has been traced very well through experiments. Bacteria will swim for about 1 second in a straight line. After that, it will tumble and it will rotate and take a diversion of about 60 degrees, and it will move again, swim. And this way it keeps on moving in zigzag manner. So this will be the movement of bacteria. Now, when a bacteria has to swim in a smooth direction, what is normally observed is, if this is a bacteria, if there are many flagellas, what is observed is all the flagella will rotate in a counterclockwise direction. All of them will rotate in a counterclockwise direction, and this will result into all flagella coming together, and it is able to move in the forward direction. If a bacteria has to tumble, and if it has to, when it comes across a repellent, what happens is, this bacteria, all the flagella will rotate in a clockwise direction. It will rotate in a clockwise direction, and it will all be scattered. When it gets scattered, it is not able to move forward and the bacteria tumbles. This is the phenomenon that has been seen. The biggest question and also the simplest one scientists asked regarding the movement of flagella is, what is moving? How is the flagellar movement occurs? So there can be two kinds of movements. One is flagella can rotate in this manner, up and down. It is kind of a wavy motion. And the second possibility is the whole flagella can rotate like this. A second kind of movement. So in order to verify, a simple experiment was carried out. This flagella is made up of a protein known as flagellin. Flagellin is a fibrous protein with about 53 kilodalton is the molecular weight of this flagellin protein. So antibodies against this flagellin was made. And then this flagella was fixed on a microscopic slide. It was fixed. Then the conditions were created in order to see how the bacteria moves. Since if the flagella is fixed on the microscopic slide, if the bacteria went on rotating, that is how they came to know that there is a kind of a motor present at the base of the flagella. Let us first of all look at the structure of this flagella and then we will look at the motor of this flagella. If you look at the structure of this flagella, interestingly, if this is a bacterium and this is the flagella. If the flagella is, as we have seen, it is composed of a protein, fibrous protein called flagellin. Now this flagellin is produced inside, and if this flagella, which is in fact, it has got a diameter of about 15 nanometer, and the inner portion is it is a hollow core, and this protein flagellin which is produced is not added here at the base. It is transported through the hollow core and it is added here. So that is how the flagella grows to the size it is supposed to be. An interesting way of constructing a molecule, very rare, unique phenomenon which we find in nature. Now, if you look at the structure of this, the filament, this is the filamental portion of the flagella. Now the filament is, it is a left-handed helical structure. It's a left-handed helical structure which is made up of this flagellin, which is a protophyll forms. They are arranged 1, 2, 3, 4 like this. They are arranged, about 11 of them are arranged. And that way it forms a kind of a protofilament. That is how the filament is formed. Now let us look at the basic structure of this motor, and the motor is at the base. So there are three important portions. One is it has a rod, it has a hook, and it has this filament. These are the three portions present in a flagellar motor which is helping to make the bacteria move in a particular direction. Let us look at the structure of this motor. This is a very, very brief diagram of the flagella. You can see that this is the filament and this portion is called the hook and this is the rod. These are the three areas of the flagella. And you look at the several membranes. So this is the outer membrane. Now the outer membrane has got a ring which is known as the L ring. Then there is a membrane here which is all becoming part of the peptidoglycan layer. Up to here will be the peptidoglycan layer. Peptidoglycan. And this layer has a P ring. Then in the cytoplasmic membrane has got It has a collection of proteins. Let us look at these important proteins which forms in the base of the flagella as well as it functions as the motor. Now, this has three important proteins. Which are the three important proteins? One is a protein called MOT-A. MOT-A protein has it has four transmembrane region. If this is the cytoplasmic membrane, it has got, this is the N terminus, one transmembrane, second transmembrane, third transmembrane, and the fourth transmembrane region. It has got four transmembrane region, which is very important for it to function as to help in the transport of electrons. Now, it also has got a cytoplasmic domain. If you look at the next protein is called the MOT-B protein. MOT-B protein, most part of the MOT-B protein is the periplasmic space. And MOT-B protein has, in the membrane, it has only a single transmembrane helix. Only a single transmembrane helix, and the remaining portion is in the periplasmic space. Very important in the case of MOT-B is, MOT-B has got an aspartic acid which is located at the 32nd position. That is very important for mobility or for the motor to function. Other important proteins in the motor is a kind of protein which is called, which forms part of what we call this is called the MS ring, that is called Fle-G protein. And Fle-G proteins, all these black ones, there are about 30 of them arranged in a circle. And surrounding that, you will have MOT-A and MOT-B protein. MOT-A and MOT-B will remain together. Like this, there will be 11 MOT-A and MOT-B proteins together. They are surrounding this MS ring. MS stands for membrane supramembrane ring. Other than this Fle-G protein, there are other proteins also present, like Fle-M protein, Fle-N protein. All these are proteins associated with the motor portion of the flagella. Let us try to understand how exactly the motor starts moving. Now remember, this motor portion that is made up of the Fle-G protein is capable of rotating. And when it rotates, this rod rotates, the hook rotates, and the filament rotates. So how exactly the Fle-G or the MS ring is rotated? So MS ring will be rotated by transporting protons, protons across this membrane. So with the movement of protons from the periplasmic space to the cytoplasmic side helps the motor to rotate. Let us look at a detailed structure of this particular MS ring. In the MS ring, the diameter of the MS ring is about 45 nanometer is the diameter. Now the 30 Fle proteins can be arranged like this. Like this, you have to arrange 30 of them. And drawing only few, there will be 30 of the Fle-G proteins. Fle-G proteins, 30 of them, which will act as the motor. In this, the hook is placed, or the rod is placed in this. Now, surrounding that, you will have 11 MOT-A-B proteins. This is MOT-A and B combined. 11 of these are arranged around this. 11 are arranged. So, if you look very closely at this combination of MOT-A and MOT-B protein together, it has two half channels. Half channel means, if you consider this is the collection of mode A and mode B together. Half channel means it opens only halfway and it opens from the other side. So there is an opening only up to here. It opens only up to here and it stops here, it blocks here. In the second half channel opens here and it opens to this end. And you see how though they are blocked, yet they can be opened. How they can be opened? Because if the aspartic acid 32 is aligned exactly here, the aspartic acid 32 is aligned exactly here. So using that, protons can be pumped across the membrane. So protons will come here, it is added to an aspartate, it becomes an aspartic acid and then it is passed across the membrane through a series of transportation, proton transportation mechanism. Now, by this transportation, so let us represent either two half channels. One half channel will allow H plus to come inside, then it rotates, it will allow H plus to go out, that result into a rotation, that movement allow the Fleege protein to rotate. So in a way the Fleege protein rotates, along with that the rod rotates, the hook rotates and the filament rotates. Bacteria are capable of transporting about 1000 protons across these half channels per second. They can transport 1000 protons per second, which can induce about 60 revolutions per second. That is about 3600 revolutions per minute. A highly efficient motor system. So this is how the motor protein operates in bacteria. How did they prove that aspartic acid 32 is very important for transportation of protons? What they have done in research is aspartic acid is replaced with, first of all it was replaced with glutamic acid. Glutamic acid, as you know, it is also an acidic amino acid which is capable of ionization. What was observed is if the motor functioned, but the efficiency was very low, stating that aspartic acid is very essential for the motor to function. A second experiment was carried out. They mutated this aspartic acid. If the motor stopped functioning. Another experiment was carried out. That is, replace the aspartic acid with asparagine. Replacing that with asparagine also result into motor not functioning. These were some of the experiments carried out in order to prove the exact role of aspartic acid 32 in transporting protons across the half channels that will result into movement of the MS ring or the Fleege protein, ultimately resulting into movement of bacteria, motion of the flagella. If you can easily calculate the energy produced by this movement. So, a simple equation, that is the proton motive force, PMF. The proton motive force is the totality of the electrical as well as the chemical potential. Combined together, that results into proton motive force, chemical as well as electrical. Together, that gives rise to the proton motive force. This can be calculated, that is PMF, this is equal to Vm minus Z delta pH. Very easy to calculate. Here Vm stands for membrane potential. Membrane potential Z is equal to 2.303 RT by F. Okay, minus 3.303 RT by F. R is the gas constant, T is the absolute temperature, and F is the Faraday's constant. It was calculated that for a pH change of 7, minus 170 millivolt is produced by the flagella in the transportation of electrons. So these were some of the experiments carried out in order to understand the rotary nature of the flagella. Please understand, we looked only from the rotary action of the flagella. We have not looked at how the signal is transducted in order to either to make it a clockwise rotation or an anti-clockwise rotation.